So in this video, we're going to be looking at how an action potential spreads along the surface of a muscle cell. Now recall back to a previous video, we did talk about how an action potential travels across a neuron. The action potential will travel across the surface of a muscle cell, but just a little bit differently. I think a lot of students get this a little bit confused. They think that the action potential is only traveling along the surface of a neuron, but that is not the case. An action potential travels across a neuron, and it also travels along the surface of a muscle cell. When we talk about the cardiovascular system, we will look at how the action potential of the SA node or the pacemaker functions, and we're also going to look at how the action potential of the myocardium or the muscle functions. So both the nerve and the muscle has an action potential, not just the nerve. So this is an amazing image that really shows how this action potential travels along the surface of the cell and causes all of these actions. So let's start at the beginning. Step one up here, this is where we can see that nerve innervating the muscle cell. So what we're looking at here is the muscle cell inside. So before we get into the muscle cell, I just want to orient our location a little bit so that you're comfortable with where this nerve is actually coming into the muscle. So this image shows the entire skeletal muscle and then all the way down to its small muscle fibers or cells. When we say muscle cell, we also mean muscle fiber. That term is used synonymously. So let's start broad picture here, looking at the entire skeletal muscle. So this would be like if we took a cross section of an actual muscle on a cadaver. This is what we would be looking at on the inside. So the epimysium is a connective tissue layer that surrounds the entire muscle itself. This is the epimysium. As we can see inside the skeletal muscle itself, we have these bundles. These are called fascicles. This entire circle here would be a fascicle, which we see here now. So this would be one of these blown up. And surrounding the muscle fascicle or the bundle of cells would be the permysium. Peri means around, permysium. Now inside the muscle fascicle, we can see more circles inside here. These are the muscle cells or the muscle fibers themselves. So around the muscle fibers, now we're at the actual muscle cell. This is what we're going to be looking at mostly with this muscle physiology. Around the muscle fiber, we have endomysium. Endo is always going to be the deepest layer. And inside this endomysium, we have capillaries as well as nerves. So both the capillaries or blood vessels and the nerves of a muscle are found in the endomysium, directly surrounding the muscle cell. So when we look at that last image, we're looking at the whole muscle cell or muscle fiber, and where that nerve connects with the muscle is in that endomysium. So let's go back to that image. All right, so this image is looking at the muscle cell, and this would be that endomysium surrounding the muscle cell or fiber, where we have that neuron coming to attach to the surface. So this is going back to that action potential of a neuron I talked about in the previous video. So it's really tying all of this together, but here we have the action potential traveling along the neuron. And once it reaches the synaptic bulb with those synaptic vesicles filled with neurotransmitters, that's going to cause the release of these neurotransmitters. These neurotransmitters will be released from that synaptic bulb, and they will float in this synaptic cleft that's in between the neuron and the cell itself. So the neurotransmitters come into the synaptic cleft, and then they're going to bind to these purple receptors here that are sitting directly on the muscle cell. That's going to trigger an opening. This is going to cause an opening of those channels. These are ligand-gated channels, meaning they will open when something clicks onto it. So sodium is going to flow in to the cell via these channels once that acetylcholine neurotransmitter binds to the ligand. So sodium flows into the muscle cell. Recall back, When a positively charged ion comes into a cell, that's going to cause it to become more positively charged, and that's going to depolarize the cell. So now we have the depolarization of the muscle cell. The action potential is going to travel along the surface of the muscle cell that we can see in these red arrows here. So action potential travels along the surface of the muscle cell. Don't get that confused. The action potential does not come into the muscle cell, but along the surface of it. When that action potential dips down into these transverse tubules or T-tubules, these are just invaginations of the membrane of the muscle cell. So action potential travels along the surface, dips down into these transverse tubules. That is going to cause this sarcoplasmic reticulum, the organelle inside the muscle cell that stores calcium, to release that calcium. So that action potential travels down into the transverse tubule and it tells that sarcoplasmic reticulum to release calcium, which we see here. Calcium is very important for muscular contraction. It can't occur without it. So calcium flows out of the sarcoplasmic reticulum and it comes down to the sarcomere. So here we are looking at a sarcomere. This is consisting of the contractile proteins that are responsible for the shortening and contraction of a muscle. So let's talk about this sarcomere first. As we can see, we have these zigzag lines on either side. These are called the Z lines. Between the Z line is defined as a sarcomere. This is one sarcomere, and a muscle has hundreds and thousands of sarcomeres, but this is just looking at one blown up. So we have Z line to Z line, sarcomere. These yellow, small, thin protein filaments are called thin filaments, also known as actin protein molecules. This is actin. The thicker filament that's in red here, which is more in the middle, this is called myosin. The thick filament is called myosin. These proteins are going to connect to one another and cause that sarcomere to shorten and thus cause the contraction of the muscle. But it can't do it without calcium. So how does calcium help this? Calcium, as we can see, floats down onto that thin filament, which we can see blown up here. So here we're looking at the thin filament, which is consisting of actin, as well as two other proteins called troponin and tropomyosin in brown. So just to clarify, the thin filament consists of three proteins. It is consisting of actin in yellow, tropomyosin in brown, and troponin in that blue teal color. So the thin filament has all three of those. And all three of those are important for the contraction of the muscle. When a muscle is at rest, which we can see here, there's no calcium binding to the troponin. We can see that these myosin binding sites, these dark little circles on the actin, are covered. They're covered by that tropomyosin protein. Those dark circles, those myosin binding sites are covered. So this means that myosin and actin cannot contract the muscle. They can't communicate, they can't attach, so there's no muscular contraction in this case. However, as we can see here, those myosin binding sites are now uncovered. So now myosin can bind to those binding sites and pull the actin towards the middle and shorten that sarcomere. Now, how does it happen? Calcium has to bind to this troponin molecule. When it binds to troponin, troponin will communicate with that tropomyosin, telling it to click off of those binding sites. So as we can see, tropomyosin is not on top of those actin filaments, not on top of those binding sites. So now those myosin little globular heads can click onto these binding sites and drag that actin filament towards the middle and shorten the muscle. So to review that, calcium released from that sarcoplasmic reticulum because of that action potential floats down to the sarcomere and binds to troponin. Troponin then tells tropomyosin, hey, we're about to contract this muscle, so hop on off those myosin binding sites, which it does, and now those myosin binding sites are exposed. And then myosin can click onto the actin and shorten that sarcomere, which we can see here. So myosin attaches to the actin, drags that actin filament or thin filament towards the middle, and that's going to shorten the sarcomere. So looking at these two sarcomeres a little bit, as we can see, this one's much longer, there's less overlap. There's less overlap of the thin and the thick filament, right? It's just a small section. But when that muscle is contracted, the overlap of the thin and thick filament is really large. It's like the whole size of the myosin or thick filament here. So what's important about this is that neither the thin nor the thick filament themselves are shortening, but the entire sarcomere is shortening because there's more of an overlap. So this actin or the thin filament is not changing shape or size. It's not changing size. Myosin, not changing size. It's just that they're now more overlapped, and that's causing the shortening of the muscle. And essentially, whenever the muscle is relaxing, we're just reversing all of this. So calcium is going to be popping off of troponin, put back into the sarcoplasmic reticulum, and then that tropomyosin will cover those myosin binding sites so that the muscle can't shorten. There's also an enzyme called acetylcholine esterase. This is going to pick up that acetylcholine in that neurotransmitter and recycle it so that there is no more action potential. So hopefully that helps you understand the muscle physiology a little bit more and how an action potential from a nerve can affect the muscle cell.